Many globin proteins from different sources like mammals, insects, plant root nodules had been determined for their three-dimensional structures independently and compared each other. The interesting thing is they shared different levels of homology in terms of their residue sequences from 16% to the 99%. However, all those globin proteins just adopt almost identical structural features as globin proteins. This observation leads to two basic questions regarding protein structures. First, how can amino acid sequences that are very different from proteins that are very similar in their three-dimensional structure? Second, what is the mechanism by which proteins adapt to the mutations in the course of their evolution? Globin proteins is pretty good to answer to those questions, basic important questions. At the time, 9 proteins were compared for their primary structure and tertiary structures. As I told you, their tertiary structures are highly conserved during evolution, while their primary structures are pretty much buried. So this is conclusion. Among about 130 long stretches, 59 positions are conserved. This could be classified into two categories. First, 31, the residues are localized between helix and helix interactions, mostly hydrophobic residues. And second group, they just contact with heme. Okay, so basically hydrophobic residues again between helix to the heme in this case. So probably this kind of interactions might key element in making globin fold. So unless mutations occur, this kind of highly conserved important key residues, proteins just could adapt globin fold. Hydrophobic interactions are very important in making proteins. This is another example for that, sickle cell anemia. There are, as you know, the hemoglobin has 2 alpha and 2 betas. Sickle cell anemia has a mutation, single mutation, so that beta subunit, glutamine at 6 position is replaced by valine. So this mutation just create hydrophobic patch on the surface that was originally negatively charged polar group. By chance, this newly replaced valine has fitting surface on other area of hemoglobin. They simply could interact with each other by this hydrophobic interaction. That force is just enough to produce polymeric forms. You see, side view of polymeric form of clustered hemoglobin. This is cross-section view for such a structure. This kind of fiber type of hemoglobin leads to the sickle cell anemia. It turns out recent years they could kill malaria parasite because the distortioned cell membrane of RBC just allows more permeability to potassium, which is abundant inside the cells, leading to low concentration inside the cells. High concentration of potassium in cytoplasm is required for malaria parasite to multiply. That's why malaria is very limited in proliferation in this patient.